going to be sort of switching gears into a slightly different topic today. But in making that switch, I want to take a second and think about a couple of the things we've already talked about. Because you just had a whole exam on different aspects of immune responses. And you might say, well, we've learned a bunch of stuff already. That should be pretty good. Why does there have to be more? And so I want to spend a second kind of thinking about places where the immune responses as we've currently learned them would be insufficient. So we can start to understand why this next set of responses is so important. So one of the things that we have learned about thus far is the action of phagocytes in the innate immune system that can phagocytose a microbe and then destroy that microbe, which is what's being shown here. And so you might say, well, it should be pretty good. Why do we need more things? One of the reasons why, in fact, we need more things is due to the fact that microbes are not just sort of passively dealing with this process. There are many of them that have evolved so that they aren't destroyed if they get phagocytosed. And so 3 different examples of what that evolution can look like are shown here. So there's one very famous organism. This is Listeria monocytogenes, not that it matters to you which organisms these are, that actually gets in the phagosome where we would like to degrade that. And this bacteria says, gee, thanks for giving me a nice home full of nutrients and stuff. And it actually breaks a hole in the phagosome and escapes and lives in cytoplasm, happily ever after. In that case, that phagocytosis process is not going to be helpful for us. There are other microbes that actually modify cell signaling so that that phagosome either can never fuse with the lysosome or so that the phagolysosome is actually not toxic and not able to do degradation. And so in both of these cases, the microbe is basically able to live happily ever after in that compartment. And these are the strategies used by Salmonella and Mycobacterium tuberculosis. And so in all of these cases, you can see that phagocytosis is not actually helping us. The microbe has figured out some way to get around being destroyed by phagocytosis. And in all 3 of the examples I'm showing you here, the microbe will live happily ever after inside of that phagocytic cell. We've also talked a bit about antibodies. The examples that I'm going to show you in thinking about antibodies are all going to be examples that kind of assume that the antibodies are neutralizing, though of course, as you are well aware, antibodies can do more than just neutralize. But we're just going to deal with neutralization examples now for ease of our life. And so here you can see an example of some antibodies neutralizing a virus so that that virus is no longer able to interact with the receptors on the host cell. This is a cartoon image of one virus, HIV. If we were going to have HIV be neutralized by antibodies like what we see on the left, those antibodies would likely respond to this yellow protein, GP120, because it's on the outside of the virus. Antibodies are proteins secreted from cells. The antibody is not going to get into the middle of the virus. The antibody is going to recognize parts on the outside, which is what you can see on the left. What that means is if you look at this virus, there are a whole bunch of parts of this virus that are basically not subject to antibody recognition in a true response. And we can deal with some other details of that and complications of that later. But in a simple sense, basically the antibodies can see this yellow protein, maybe some of this gray stuff, maybe a little of this red protein, but all this stuff inside is completely hidden from antibodies because there's a membrane and there's a wall around it. This would be sort of like if you went to the airport and showed someone your suitcase, and they said, that looks like a nice suitcase. It must not have anything bad inside it. Go ahead. Right? That's really what we're doing, is we're looking at the outside with the antibodies, and we're not looking at the inside at all. And as the way some of you giggled when I gave that airport example, you know that that might not always be the best strategy. And so we're basically not seeing a whole bunch of information. We're not getting a whole bunch of information to help us learn about this virus. Viruses take advantage of this. They can, in fact, often do things like shift parts of the outside of their capsid while leaving all the important stuff unchanged. So we could have this virus that starts out with a purple protein on its surface, and then when we have purple antibodies around, the virus can just switch and use a red protein, but leave everybody else in the middle unchanged. And do the same as that antibody response proceeds. And so if we just had antibodies, you can see that microbes could get around us pretty easily. They could foil us pretty easily based on this. And so the problem that we see here, or here, is that we're only really looking at the outsides of things. We're not really looking at the insides of things. The one other big complication also is something that at least I think about when I think about viral infection. Surprise, I think about viral infection a lot. I know everyone's shocked. So this is an example of a virus replication cycle. So this is an example of HIV's replication cycle. If this were my virology class, I would go through lots of steps. I see all sorts of steps. We don't really need to worry about the steps right now, other than to note that the first step is the virus going from outside to sort of inside. And then there's a whole bunch of inside the cell steps, and then the virus leaves again. And if you were to sort of time this, most of the time, the virus is inside and not outside of the cell. Now, if we think about our antibodies, antibodies are going to be able to attack this virus outside of the cell. But antibodies aren't coming into our cells to look in the cytoplasm. They're basically largely looking outside. Again, there is some novel, cool information about intracellular antibodies. We're going to ignore that right now. So most of the time, we can't actually see this virus either because it's inside of our cells. And so we have this problem of we're not doing any surveillance for things inside of our cells. That's most of the time of a virus's life cycle. You can see that we had some bacteria that were inside of the phagocytes. You can see that we weren't able to see the inside of our microbes. And so all of these are ways that what we've learned thus far about the immune response are insufficient. They don't cover all of the things we need to defend against. And so as a result, we also have this other arm of the adaptive immune response, which is the T cell arm. And so T cells are going to be able to solve the problems that I showed you today. So all of the types of immune responses where I said antibodies or phagocytes aren't good enough are actually all examples of places where T cells are super useful. So T cells are type of lymphocyte. So you can see that they develop just like the B cells do from a lymphoid precursor. They're part of the adaptive immune system, and there are lots of different types of T cells, which we will see more of. that actually makes it really hard to define T cells because there are multiple types of T cells that each do slightly different things. I've only ever heard one definition that someone gave that tried to define all of T cells. And so this one definition that I've heard that defines all of T cells is that T cells interact with other cells, other body cells, and make them change. And that's kind of vague. Here you can see kind of the two big groups of T cells, though we will see more and more of them throughout the semester. One type of T cell is really good at producing cytokines, which can impact every other cell of the immune system's response. And so that cell is really going to be able to impact and change all the other kinds of responses of pretty much every other cell of the immune response. We usually refer to those as helper T cells. There are other T cells that actually are going to be able to kill infected cells. And so these are known as our cytotoxic T cells. And so these are kind of big picture what T cells do, how they work. We are going to see much more about T cell function later on. But in order to think about T cell function, we need to think a little bit about things like how the T cell is activated and the antigen that T cells are seeing. So this image kind of is making a compare and contrast between our B cells and their ability to make antibodies and our T cells and their actions. So this is sort of what lymphocytes do or what adaptive immune cells do. We can also do a little compare and contrast of receptors. If you remember, when we learned about B cells, they had a specific receptor. It was very creatively named the B cell receptor. The B cell receptor, if you remember, was an antibody with a transmembrane domain. And so oftentimes when we think about B cell receptors and antibodies, we think about them pretty interchangeably. So this shows an antibody, but you can imagine it as a B cell receptor. The T cell also has a very creatively named receptor on its surface called the T cell receptor. And the T cell receptor is shown on the right. What you can notice is that the T cell receptor has two chains, two different protein chains. They are made up of immunoglobulin domains, so you can see the rectangles. The two chains are held together by a disulfide bond. We've got a very short transmembrane domain and a little tiny cytoplasmic tail, but it has all the same problems that the B cell receptor has in terms of having a little tiny cytoplasmic tail. And both of these T cell receptor chains contain a constant region and a variable region. The variable region has kind of three little pieces that come out and stick out and touch the antigen. And in fact, for pretty much most of our purposes, the T cell receptor is structurally almost identical to an FAB, to one of these antigen binding arms, in terms of being two proteins held together by a disulfide bond. We actually do call them heavy and light chains, though. Again, that's a problem for later. With hypervariable regions that are binding antigen, made out of immunoglobulin domains. It's really similar to this structure, just with a transmembrane domain. But again, the trick comes in to sort of exactly how and what this T cell receptor is binding. And so immunologists started to really understand this problem with a very famous experiment done by two scientists, Zinc and Doherty. And so I want to explain the Zinc and Doherty experiment. This is yet another of the Nobel Prize winning experiments that we talk about this semester. And in this experiment, Zinkernagel and Doherty were trying to understand some details of T cell recognition and how the T cell receptor worked. In this experiment, they were using mice of two different strains. One is here called strain A. One is here called strain B. When we talk about strains of mice, a couple things you want to be aware of. So a strain of mouse is sort of a genetically defined mouse lineage. What we do when we're breeding those mice is that we actually have a whole bunch of genetically identical mice that we're breeding together. So every mouse of strain A, you can imagine, is genetically identical to one another. Every mouse of strain B is genetically identical, but different than A. So if I say something is a mouse strain, it means it's sort of every mouse of that same strain is genetically identical. We often consider them like repeats of each other for sort of replicates for experiments. You know, it's copies of the same individual. So they did use mice here of strain A and strain B. You can think of that as two different genetic types. And the first thing that they did was they infected a mouse, and this mouse was the genetic type strain A. And they infected this mouse with a virus. You don't really care which virus it is. It's called LCMV or lymphocytic choriomeningitis virus, whatever. So they infected a mouse, a strain A mouse, with LCMV. And they waited to allow T cells to be able to make their response. And specifically, this experiment was looking at the killer type of T cells, the cytotoxic T cells. So they waited for 7 days, and then they took those T cells out of the mouse. They took the spleen, they got the T cells out of the mouse. So here's our T cells. And they wanted to test the ability of those T cells to act against other cells. And they used a lot of different other cells. This shows 2 of them, but I'll actually mention a few different ones. So one of the things that they did is they took either some cells from a strain A mouse or some cells from a strain B mouse, and they infected those with the same virus, LCMV. So these are cells from another mouse, either genetically identical, also strain A, or a different genetic type, strain B, infected with the same virus. In the future, we will also see some that have a different virus or some that are not infected with any virus. So they infected those, and then they mixed them together in a dish. And their goal was to see whether these T cells could kill these infected cells. That was kind of what they were trying to do here. These infected cells that hopefully would get killed in this experiment are called the target cells. And the way that they did this experiment, the way you actually do this experiment, is that you take your target cells and you fill them up with radioactive chromium, so chromium-51. Honestly, all you have to do is just grow the cells in some media with it, and they take it up. And so you get a whole bunch of radioactivity in the cells, and you wash away the excess radioactivity, so you just have radioactive cells. And then you put the T cells with those radioactive targets, and you look to see if the T cells can kill the radioactive targets. And if they can, all the radioactivity leaks into the media. And so you basically just look, did I have radioactivity in the media or not? Did it leak out of my cells or not? Did my T cells actually kill? This is called a cytotoxicity assay. So they used a cytotoxicity assay, and their outcome was either they got killing or they got no killing. in their experiment. So these cells, the target cells on the right, are the ones that are loaded with radioactive chromium. And we're looking at the ability of these T cells from a strain A mouse infected with LCMV to kill those targets. And so here are some of the outcomes of this experiment. When they used T cells, which came from a strain A mouse infected with LCMV, and they used target cells from a strain A mouse infected with the same virus, then the T cells could kill the targets. So same genetic type, same virus, we get killing. When they use a strain A mouse set of T cells and target cells from a strain A mouse that were not infected with a virus, no killing happened. So that makes sense. There's no virus here. You don't really want your T cells killing all the cells of your body. That would be bad. Notice you are still alive and you still have cells. So the genetics match, but we don't have the virus, and we don't get killing. Not shown here is the fact that they also did situations where they had some strain A target cells that were infected with a different virus. So we'll say flu. And if they did strain A T cells that came from a mouse infected with LCMV and strain A target cells that were infected with flu, no killing. So they saw this was specific. Had to be the right virus. So the virus had to match. These came from an LCMV infected mouse. These were LCMV infected. But the other really important piece of this is they took these T cells. They came from a strain A mouse that was infected with LCMV, and they used target cells from a strain B mouse that was infected with LCMV. So we got the same virus here, but these cells are from two different genetic types. And in that case, the T cells didn't work. The T cells could not kill the targets. And so if the genetic background of the target cell and the killer cell didn't match, the T cells didn't work. This is very different than what we saw with antibodies. So remember, we talked about the fact that sometimes we can take antibodies from a person who survived an infection. I can just pull some of those antibodies out of you and put them into me, and that will protect me. Your antibodies work just fine against my virus. But here, T cells don't seem to work the same way. They seem to care about the genetic background of the cell they're trying to kill. And this led us to realize that T cells can't just recognize antigen. It's not just about the virus. There's also some other host protein that must be involved. That's part of this whole genetics thing. There's something besides the antigen that's involved. And so what we now know is that while B cell receptors just binds to antigen and there's nothing else involved, the T cell receptor binds to antigen along with this other self-protein, which is known as MHC. So what is going to happen here is that we've got all this inside stuff, things from the inside of the virus, things from the inside of the cell that are getting shown off on the surface of the cell on like a little platform. And the T cell receptor sees both the platform and the antigen peptide. Both of those together make up the antigen for the T cell receptor. And so if we have the wrong platform or if we have the wrong peptide, the T cell won't work. So that protein, that sort of platform that I've mentioned, is referred to as the MHC. And this leads to a phenomenon known as MHC restriction. So T cells are what is called MHC restricted. That means that T cells can only respond to some MHCs. They can only respond to the right MHC. They can't respond to the wrong MHC. They're restricted to only being able to work against the right one. So here we can see a T cell with a T cell receptor. And it is binding to one kind of MHC, which is this yellow MHC, that is presenting this antigen, antigen X. And so we've got the right MHC and the right peptide. The T cell recognizes, and the T cell is going to be able to do its T cell stuff. In the case, over here on the right, we've got our T cell receptor. You can see we've got the correct MHC, but we have the wrong peptide. So maybe this is from a different virus. This doesn't match now. We can't have this MHC be recognized by this T cell. No recognition, T cell doesn't act. But also, if we have the right peptide from the virus, so this is LCMV, but with a different MHC from a different genetic background, the T cell will not recognize this. So we have no T cell recognition. The T cell will not work in this case. So there's this additional piece that comes in with T cell recognition. There's one other piece that I have slipped and said a couple times while I was describing this, but haven't officially told you, so I will now officially tell you. MHC is only able to bind protein. And so T cells only recognize proteins. T cells do not recognize nucleic acids, lipids, or carbohydrates because MHC cannot show, present is the term. MHC cannot present lipids, carbohydrates, or nucleic acids. MHC can only present peptides, and so T cells only recognize peptides. And that is different than antibodies, which can bind to any type of biological molecule. And so the function of MHC is sort of shown in cartoon form here. We have some kind of protein from our microbe in our cell. That protein is going to get degraded by proteolysis. And then some pieces, some peptides of it will be shown on the surface of the cell on MHC. And then the T cell will sort of come along and look and see what's going on. Do I match any of those things? When Dr. Miller talks about this with her non-majors, microbiology students, she tells them about MHC as being like a little kindergartner who's like, Mommy, Mommy, look what I found. You know, showing off their trophy. I like to think about it a little bit differently because there's one other complication I have to teach you about later. And so I think about it as more of a way that the cell is showing off what's inside of it and saying, am I okay? So basically the cell is taking whatever it's got inside and saying, am I okay? Is this okay? Can you look at this and see if this is okay? And so it's not even necessarily like the cell knows what it caught. The cell just is like, is this stuff fine? Is this stuff I got okay? So that we can see those things that are internal. On Friday, we're going to spend a lot more time talking about the specific details of the MHC molecule and its sort of specific biochemical structure. And then next week, we're going to talk about kind of the cell biology of the rest of this cartoon, of how we get from the microbe to a thing on MHC on the surface. So don't worry, we are going to talk about that stuff, just not right now. Also, when MHC is doing its presentation, it is always presenting what are known as linear epitopes. So remember that with the antibody, we could think about whether the antigen was folded or not. In the case of MHC presentation to T cells, the antigen always gets cut up, and we are always presenting some little stretch of the peptide that can be found in the primary sequence. It's just a linear stretch of amino acids. There's no folding involved. So we always have these linear epitopes. They could be buried in the middle of the protein. They could be from any different protein of the whole microbe. So we got lots of choices for the antigen. And the epitope will be some short peptide that is actually bound to the MHC and then put on the surface of the cell. You can actually see this in more detail here as well. For some reason, this figure really clicks for me. You're going to see this figure a lot because it seems to make sense for me as a way to explain some things. And so here you can see an MHC molecule in green and a T cell in pink, or a T cell receptor, and a peptide being presented in brown. And what you can sort of notice is that the MHC has a packet that holds the peptide. And so that's sometimes known as the peptide binding cleft or the peptide binding groove. And some parts of the peptide are going to be interacting with the MHC molecule, while other parts of the peptide are going to be interacting with the T cell receptor. And you can see that the T cell receptor is actually making contact both with the MHC and with some parts of the peptide. If we kind of look at this in a slightly different way, you can see some of the structure parts of this here. So you can see, so at the top, this is a T cell receptor, the constant regions and the variable regions. This is the MHC with the peptide in yellow. And what you can notice is you've got these little loops coming down off of the T cell receptor, making contact with the MHC plus peptide. They're the same loops you saw with the antibody. And so basically, you're just looking at the same TCR plus MHC in a different format here. Those are the hypervariable loops, or the CDR loops, that are making those contacts. Here you can actually see if you were the T cell receptor and you were looking down at the peptide plus MHC, the gray is the MHC, this little yellow in between is the peptide, and you can see that you're going to that T cell makes contact all across that surface. It's really kind of just like a surface as far as the T cell is concerned. And our three different CDRs are the things that are really going through and touching. And in fact, we even know that there's a specific angle, which is this gray box. The gray box is the T cell here, that the T cell tends to interact with the MHC at. And in fact, there are some cases, like in some autoimmune diseases, the problem is that the T cell and the MHC are actually at the wrong angle. So we even know about the correct angles happening here. But largely for the rest of the time today, I think everything else I have to say today is basically this. Yeah, it is. I want to talk a little bit about some aspects of MHC genetics. And again, I will say some more about MHC genetics a bit on Friday as well. But I want to kind of think about the big picture of MHC genetics here. We're going to be thinking about some MHC genetics stuff and some nomenclature a little bit today, as well as a bit on Friday. This is one of those places where immunology nomenclature gets a little obnoxious. Because people who were studying this in humans found these weird proteins that were in the serum of pregnant women, and they were just like, look at these fun proteins on their white blood cells. They were called the human leukocyte antigens. And they were on some leukocytes in pregnant women, and they were like, this is great. And they just were doing some experiments with them. And there were some other people who were doing some experiments with mice, giving mice skin transplants. And they didn't know they were all working on the same proteins. And so there's a set of names for things in humans, and there's a set of names for things in mice, and they're sort of different. Today, for all of my examples, I'm going to give you the human names. I will go through the mouse nomenclature on Friday, because we see it in so many experiments that you have to kind of have some familiarity with that. Like every paper is going to have the mouse nomenclature in it. But today, all of my examples will use the human nomenclature. No matter which species we're talking about, we can refer to these genes and the proteins they encode as the MHC. There are special names for human MHC or mouse MHC, but everybody's is called MHC. And no matter which species we're talking about, there are 3 general regions of the genome that are sort of part of the MHC region of the genome. One thing that's sort of cool is that all of these genes are together in the genome, on the chromosome. In the case of humans, this is actually the entire short arm of chromosome 6. And the way that so one thing that also ties into MHC is really important for transplantation. So if you're going to do any kind of transplanting, you know how you have to do some matching of genes? Yeah, it's MHC that you're worried about. You're just worried about MHC matching. And the reason why I can always remember that the human is on chromosome 6, and it's the entire short arm of chromosome 6, is because Michael Crichton wrote a book called Chromosome 6, where they were making monkeys that had human short arm of chromosome 6 so they could be used for human organ transplants. And in the book, he said that chromosome 6 also was what gave you sentience, and so the monkeys became sentient and could use. Anyway, it's a really wacky book. It's real wacky, but it's called Chromosome 6, and so that's how I always can remember. This is chromosome 6. But no matter which species we're talking about, in their MHC gene region, there is a class 1 region, a class 2 region, and a class 3 region. The class 1 region contains the class 1 MHC genes. I know, shocking, right? The class 2 regions contains the class 2 MHC genes. Right, mind-blowing. The class 3 region contains a whole bunch of other genes that are important in the immune system that are not actually MHC at all, which includes all of the complement genes. So all of complement, some different cytokines, it's a whole bunch of other immunologically important stuff is in this class 3 region. It's not actually MHC. But the class 1 contains the class 1s, the class 2 contains the class 2. Okay. And so specifically, you can see the human version of all of this here. So human MHC is known as HLA, or the human leukocyte antigen. So if I ever refer to something as HLA whatever, HLA means it's human MHC. So that's like code. We have the class 1, the class 2, and the class 3 region. And in human, we have 3 different types of MHC class 1. They are known as HLA-A, HLA-B, and HLA-C. We also have some human class 2 genes. They are HLA-DR, HLA-DP, and HLA-DQ. So these are the human class 1s and the human class 2s. I like to remember that class 1s have 1 letter and class 2s have 2 letters with A, B, and C, or DR, DP, and DQ. And so you can see the A, B, and C genes here in the class 1 region. Officially on the chromosome it goes BCA because Y could ever not be straightforward. You can see the DPs, DQs, and DRs in the class 2 region. You can see that there are also some other proteins that are sort of interesting that can be dealt with later. So if you think about your genetics, you have a copy of chromosome 6 from your mom, and you have a copy of chromosome 6 from your dad. And so you can see that in this cell here. We've got mom's MHC. You can see her HLA-A, HLA-B, HLA-C, dad's HLA-A, HLA-B, HLA-C from each of these 2 chromosomes. You can also see the class 2s. One of the really critical things here to know about MHC is that MHC genes are codominantly expressed. So what that means is that if we actually look at the surface of this cell will have 12 different MHC molecules on its surface. So it will have A1 from mom, B1 from mom, C1 from mom, A2 from mom, A2 from dad, B2 from dad, C2 from dad. So we've got 6 class 1s. 6 class 1s showing up here. And we've got 6 class 2s. The DR1, DQ1, DP1, DP2, DQ2, DR2, which you can see down here. So oftentimes, again, in my research past, I spent a lot of time thinking about MHC class 1. So a lot of times I'll just go with, so you know, the 6 class 1s, technically there's 6 class 1s and 6 class 2s. I just think a lot more about the 6 class 1s. Are codominantly expressed. So every single, wrong. Your cells have all of these on their surface. I haven't told you which cells have them yet, so that's cool. But there's one other piece to this that we can think a little bit about. So let's imagine for a second that I decided I was going to sequence some genes from your genome. Let's imagine that I was going to sequence all of the copies of actin of everyone in this room. So the actin gene you got from your mom and the actin gene you got from your dad. So there are 14 of you and 1 of me, so there's 15 of us total. That means we've got 30 chromosomes. How many different versions of actin do you think I would find? Yeah, Jamie. I would probably find like 1-ish. And the reason why I would find 1-ish is because actin's, it's really important that actin's kind of conserved. If you picked up many mutations in actin, you'd kind of not have cells. And so we probably, among all of us in this room, have like 1 allele of actin. Even though we got 30 chromosomes to choose from, we all probably have 2 copies of the same actin, right? Because if we had different structures of actin, and yes, we could have the same gene, same structure with different genes. We're not going to deal with that now. But if we had different structures of actin, then like we might not have cells, right? So we would imagine there's probably like 1 allele of actin, or a very small number of alleles of actin. That is not the case with MHC. So MHC genes are what are known as the most polymorphic genes in the human genome. That means there is the largest number of different alleles. These data are from January 2017, and it's actually really important that the textbook tells us that because this changes so frequently. So 5 years ago, there were 3,221 known versions of HLA-B in the human population, and that number has certainly increased. And 2,480 HLA-As, and so you can see that the numbers of alleles of these are huge. And so what that means is that we will really have a situation that looks like this. It is very unlikely that you got the same HLA-A from your mom and from your dad. It's very unlikely that you have the same HLA-B from your mom and dad. C, you really have 6 different ones, or it's most likely that you have 6 different ones, probability-wise. Because there are so many out there. And you probably have 6 different class 2s as well. And so you really do have these 12 different MHC molecules. And if we were to compare from person to person in this room, we would see that we each have a different combo. So while you and I might share one HLA-A gene in common, we all have a different combination of 12. And so we are all totally unique in terms of which 12 we have. This has big implications for kind of T-cell responses and how they work. This also has a lot of other sort of interesting genetic applications. This is one of the ways that we do a lot of genetic testing for a number of different situations, because we know that HLA molecules are next to each other on the genome, and they travel in what is known as a haplotype, which means a bunch of genes inherited together. They are, in fact, the longest haplotype in the human genome in terms of genes that always transmit together. Just to make your life slightly easier, the second longest haplotype in the human genome is the one that controls skin color, hair color, eye color. It's why you tend to see super pale people with blonde hair and blue eyes. Those genes often go together. You know that they don't 100% of the time go together. Say some people have super pale skin, blue eyes, and not blonde hair. So it's not 100% of the time that those go together, but that they often do. And similarly, these genes often are like really seen going together. And we can actually use that information for a lot of types of genetic testing. So here is one Maury Povich example. Jerry Springer, something like that. So here we can see an offspring and the specific HLA-A, HLA-B, and HLA-C types they have. We can see those listed here. We can see the mom's HLA types, and we can see the potential dad's HLA types. And so by looking at the different HLA types, we can figure out pretty easily which of these would be the dad. So what we can do is we can look at first HLA-A. Look at the offspring and the mom. Which HLA-A gene did this child get from their mom? Hannah.
The A3.
A3. How do you know it's A3?
Because there's no 11.
Because they don't have 11, so they couldn't have gotten 11, so it has to be A3. That's the only one they have in common with their mom. So then what did they have to get from their dad? 43. So then who could possibly be the dad? Potential father 2. And we can go through the same thing with B and C to sort of check our work. And so this is actually how a lot of genetic testing is done, because we do have these unique alleles in this way. We can also think about questions like this one that is from an old exam. And so here I have a mother, a father, and a child, and I ask you to identify the MHC haplotypes of the mother and father. when I ask you to do this, both mother and father have 2 haplotypes. So we got to have 4 answers. One of the most common things that people do wrong here is they give me only 2 answers instead of all 4. And what I can do is and basically another way of phrasing this is me asking you which of these genes are together on which chromosomes. So I know that the child got A2 from mom. You can see how I know that the child got A2 from mom. So one of mom's chromosomes has A2, which, like, thank you, Captain Obvious. Yes, one of mom's chromosomes has A2. The child also got B27 from mom, right? That means that A2 and B27 were on the same chromosome in mom, because she gave that chromosome to her kid. And we can also see that DR3 must have been on that same chromosome, because she gave that one to the kid too. And so we can say, well, one of mom's chromosomes has A2, B27, and DR3. That's one of mom's haplotypes. And we can say, well, mom's other haplotype must be A3, because that's the one that's left for her A's. And B12, because that's the one that's left for her B's. And DR3. I clearly got tired of making up different ones, so sometimes I repeated them when I was making this up. And so that would be half of the answer here. So then what are dad's haplotypes? Justin. A20. Okay, what goes with A20?
The child and the father.
Yeah.
So that's the first one.
Okay, Jin.
That's
So B57 and DR5. And then what's dad's other haplotype? Krishna.
A1,
Okay.
B7, and DR2.
Yeah. Full credit. Good job. And so this is one thing that we can do with MHC. We also, however, can do some other types of, get some other types of information based on all of this. So I told you already that the MHC genes are the most polymorphic genes in the human genome. But what I didn't tell you is what part of the protein changes based on all of those different genetic changes. Where are all of the changes in the protein? Where's the variability? And if we look at our MHC molecules and we look to see where A and B and C are different from each other, even though they're all class 1, or we look to see where A1 and A2 and A3 are different from each other, we find that all of those differences map to the peptide binding pocket. So basically what you can imagine for all of these MHC molecules is that they all differ in their peptide binding pockets. So all of the differences are the parts are where you see stuff in red, when you see the red dot. So it's all in that pocket that is binding the peptide. And so what that means is that in every different type of MHC molecule, this pocket is slightly differently shaped. It can thus bind to slightly different peptides. So this one binds to the one with the circly amino acid and the one with the triangle-y amino acid. And this one's at, like, I don't know, the second amino acid position and like the 6th. You can imagine a different MHC might bind to, like, hexagon in the middle, because the shape of the pocket would be different. And so each MHC is different based on the shape of this pocket, which dictates what kind of peptides it is able to bind. And so you can see this here. as an example. These are actually 2 mouse examples, but we don't care at the moment. So these are 2 different types of MHC molecules, and this shows you peptides that can be bound to each of those. So you can see that this MHC binds to this peptide, or this peptide, or this peptide, or this peptide. Basically anything that is about 8 amino acids long and has an L or an I at the 8th position, and it has an F or a Y at the 5th position. And it can actually bind to peptides from hepatitis B virus or influenza or LCMV or all these other viruses. While this other MHC type binds to a different class of peptides. So now the peptide has to have a Y amino acid at position 2. But we can make responses to peptides from all of these types of microbes. So each MHC type is going to be sort of ideal for presenting some peptides from different microbes. So my MHC, I might have an MHC type called HLA-B27. I have never had my HLA type, so I'm just picking some randomly out of the air. I might have B27. If I have B27, B27 presents peptides from HIV really well and would make me have an awesome immune response to HIV. I would not get very sick if I had HIV. But maybe I might not have the right MHC type for some other microbe. And so this ends up working out for us sort of on a population level. Someone in the population has the right MHC for every microbe. It might not be you. You might be the sad person who's not going to be okay in that situation. But as a population of humans, we're good. Because we have this dramatic diversity in our MHCs. And so this is sort of showing you an example. Different HLA types will have different impacts on what happens to you with HIV infection, whether you proceed to disease slowly or fast, whether you have a low or high amount of virus, all based on which MHCs you have. And this also gets into sort of thinking a little bit about this idea of the codominant expression of the MHC genes. Because you have 6 class 1s on the surface of your cells, that means you got 6 chances to have the right binder, or to have a good binder for each microbe. You've got 6 chances for class 2 to have the right pocket to bind something good for that microbe. So that's why it's really important that they're codominantly expressed, and why it's so important that they're all so different. Now, if you think about your kids, you probably want them to get some MHCs that are really different than yours. You know, you don't want to end up having a partner who has the same MHCs as yours, so you both give them A1, and you both give them C1, and you both give them B1, right? Like, doesn't that seem to make sense? So this is one of the craziest things I've ever heard in my life. Someone actually did a study and said, it would be really, we think that there should be a role for MHC in who you're attracted to. And it's like, that is wackadoodle. And someone actually did an experiment on this. So I'm going to show you the data. When I used to do this class in the spring, this always used to fall right around Valentine's Day. I gave this lecture on Valentine's Day multiple times. So, you know, that's fun and exciting, because we've got this very polymorphic gene, these super polymorphic genes. So what they actually did is they took a whole bunch of male and female students, and they looked at their MHC types, and they made a male student wear a t-shirt for a while. And I remember that they had to work out in the t-shirt and get sweaty in the t-shirt. It is specifically known as the sweaty t-shirt experiment. And then they gave them to the female students to ask them which one smelled the most attractive. And the females seemed to always want to like the ones that had the most diverse MHC from theirs. How in the heck that could work at a molecular biology level, I have no idea. And again, I often think of this as being a little weird. But this is one of many fun MHC facts. On a slightly more serious note, MHC has actually also been shown to vary pretty differently among populations around the world. This is largely due to probably things like founder effect and some other evolutionary effects. But in fact, anthropologists use MHC pretty often to try to determine lineage relationships between different groups of people by looking at the specifics of the MHC types of those groups of people to see, sometimes they'll find isolated tribes and not know who they are most related to, and they actually do all of those, all the work using MHCs to figure out which groups are much more closely related to which other groups. You can also imagine that this has some interesting impacts in terms of global health issues, given some of the differences in MHCs. I mentioned to you that you could have the right MHC type for a particular infectious disease. You also can have the wrong MHC types in a lot of situations. So for pretty much all of our major autoimmune diseases, they are all linked to having some MHC type. And so in general, you can imagine that there are some T cell presenting some self-antigen incorrectly. It has to be done on a particular MHC type, and so there is genetic linkage between most things and MHC. So for example, HLA-B7 or B27 is the good one for HIV, but it also has this downside of being related to an autoimmune disease. So it's not like you can imagine there's the good HLA type and the bad HLA type. They all kind of have pluses and minuses. And this is a short and very out-of-date list of diseases that have all been linked to HLA type. The answer is pretty much all the things at this point. Sometimes people do these multi-million-dollar studies of genetics to try to find like the gene important for such and such disease, and oftentimes they find MHC. And it was like an immune disease, and I'm like, thank you, Captain Obvious. We already knew that was an immune disease, and not a surprise that MHC is the gene that you get. But you can see basically here just how many different disease states are related to MHC type.